Everybody, this is Mrs. Houts, and we are going to talk about the next unit, Unit 4, which is about cell communication in the cell cycle. And we are going to start with Chapter 11. Okay, so this is just a tip about reminding you to use all those extra resources on Schoology. Somebody actually asked me before the last test if there were good videos, and Bozeman Science makes great videos. A lot of people like these Amoeba Sisters. So there's some options for you to watch the videos. And there's a whole bunch of stuff listed here too. Crash course. Lots of things to find if you want to see the material in another way. Okay, so now we need to learn about how to maintain all the cellular conditions that you just learned about. All the metabolism, respiration, all those things from the cell structure and function and how that gets maintained so that things can happen the way they're supposed to. All right, so the first word to worry about is called homeostasis. Okay, and that just means maintaining a steady state in a living thing, in your body, in a living cell, whatever. Okay, so we need to maintain near constant body conditions, which is known as homeostasis. All right, and then this example of a thermostat. If you look here in the middle, we have a thermostat. It's been set at 20 degrees Celsius. Why on earth anybody would, I guess that's probably about room temperature actually. I was thinking Fahrenheit for a second there. And then if you're in your house and you have it set at 20 and your house gets cold, right? The room is cold, you know, the thermostat's going to kick in and turn the heater on, and then the temperature in your room will go up. So we have something called a stimulus, which is the temperature of the room. And then we have a response. So in this case, the room is cold. The response is that the heater turns on, and then the temperature goes up. Okay, on the other hand, if the room is hot, okay, the stimulus is that it's too hot in the room. The response is that the thermostat turns the heat down, and the temperature decreases. So this is just an example of how a thermostat is maintaining homeostasis in your house. Okay, there's two important parts to this, or things to understand, I should say. Negative feedback is one way that homeostasis is maintained, and that is a control mechanism that will reduce the stimulus in order to maintain homeostasis. That's negative feedback. Positive feedback is a way homeostasis is maintained, and it works by turning up the stimulus, amplifying the stimulus. Oh, sorry, and I misspoke. That is a way to deviate from homeostasis. So in our thermostat example, here's our room. It's set to 20. If it feels too cold, the stimulus is that it's too cold, and the heater turns on. So in this case, the response is acting against the stimulus to maintain the temperature. In this case, the room is too hot, and the heater turns off. So again, the response is acting against the stimulus. So in both of those cases, that is an example of negative feedback. All right, remember, negative feedback reduces the stimulus to maintain homeostasis. Okay, thermoregulation happens obviously in living things as well, okay, in a way for things that are living to maintain their temperature within a normal range, okay? And this is important for one reason, is because we know that proteins and enzymes can get denatured if the temperature is not optimal for them. So for example, this red enzyme, which is amylase, works best at about 25 degrees. So if that cell that's using the enzyme doesn't maintain its temperature within that range, that enzyme's not going to function very well. Okay, and I think we've talked about this too. Other enzymes have different optimal temperatures. Okay, so in each case, you know, whatever that living thing is that's using it, or different parts of your body perhaps have different temperatures that are the best. They need to try to maintain their homeostasis so that their enzymes function properly. Okay, here's just an example of an egg, proteins of an egg getting denatured. When the temperature is too high, you cook an egg, turn up the heat, and all of these proteins get denatured. Okay, and this is just a reminder down here of how enzyme action works. Here's our enzyme. Here's the active site. The substrate binds into the active site and reduces the activation energy, and then it can do its reaction and we end up with products. Okay, so if the enzyme's not at its proper temperature, that is not going to work very well. Okay, so there's a couple different types of living things, and they have different mechanisms by which they maintain their body temperature. The first one is called an ectotherm, okay, and those kinds of living things get heat from the surroundings, okay, and their body temperature changes as their environment changes. So we used to call these things cold-blooded. All right, so some examples are all the reptiles and the fish, invertebrates. Okay, they are not maintaining their body temperature really on their own. They're using their environment to maintain their body temperature. So they are doing that using their behavior. Okay, so this is an external control of their body temperature. Ectotherm. Okay, here's some turtles laying in the sun. Okay, maintaining their body temperature by sunning. Here's a lizard in the shade over here trying to keep cool by putting himself into the shade. Okay, so ectotherms maintain their homeostasis of their temperature by their behavior. Okay, they use the external environment to do that for them. Okay, one advantage for ectotherms is that it saves a lot of energy. They are not using their own energy to maintain their body temperature. The downside is that their metabolism is a lot slower when it's cooler, right? So if they're sitting in the shade, they're not moving very fast, okay? Their metabolism slows down. Okay, so that old saying about the early bird catches the worm is true because the worm is an ectotherm and it's cooler in the morning, it's not moving very fast, so the birds can eat them. All right, this is a really awesome video. I am not going to play it right now, but I would really recommend you watch it. It's super fun and interesting. All right, another kind of animal regulates their body temperature in a different way. These are called endotherms, and they generate body heat via their own metabolism to maintain an almost constant body temperature. Okay, these are what we used to call warm-blooded, okay? Some examples are birds and mammals. Okay, birds and mammals use their own metabolism to maintain their body temperature. All right, we sweat, we shiver, our skin and blood vessels dilate or contract. Here are some pictures of those things. And we do those things and it helps us to maintain our body temperature. Okay, a positive side of that is that we can be active at any time of the day because we're not dependent upon the sun being up or the shade to maintain our temperature. The downside is that we can use up a lot of energy to stay warm. This little picture is of a little shrew, and it loses a lot of body heat because it's very tiny, and it has to expend a lot of energy to keep warm. So in this case, that shrew has to eat enough food that adds up to 1.5 times its body weight, okay, in order to have enough energy to maintain its warmth, its body temperature. Okay, and luckily for us, we don't have to do that quite the same way because we don't want to eat 1,000 quarter-pounders a day. That would be gross. Quarter-pounders. Ugh. Okay. All right. Another important way to maintain homeostasis is called osmoregulation. The word osmo, hopefully, set off some bells for you about osmosis. Okay, so this is the process of maintaining homeostasis of the solute concentrations and the water balance in a living cell. Okay, and this is a little review. All right, we have a cell shown in isotonic solution or an environment, hypotonic environment, and a hypertonic environment. So just pause for a minute, think back. What is going to happen in each of these situations? Okay, this is why it's really important for cells to maintain, their water balance, their solute balance via osmoregulation. All right, an isotonic cell means that the solute concentration inside the cell and outside the cell is roughly equal. Okay, so the net water flow in and out of the cell will be in equilibrium. Hypotonic solution means that the solution has fewer solute particles than the inside of the cell. We assume these red ones are the solute particles. There's more in the cell than outside of the cell, and in this case water is going to go into the cell to try to maintain that, to try to equalize that concentration. A hypertonic environment is when the outside of the cell has a higher solute concentration than the inside. So in each of these cases, isotonic is healthy. The cell can maintain its homeostasis. The hypotonic situation, the cell could very easily burst. Okay, that cell is going to fill up with water and it's going to burst. If it's a plant cell, that cell wall will prevent it from bursting, but it will get very full of water in what's called turgid. In a hypertonic solution, the water is going to go rushing out of the cell and the cell is going to shrink up. Okay, so it's very important that cells maintain their water and solute balance and remain isotonic via osmoregulation. Okay, another reason is that some solutes are toxic and they need to get removed from the cell. So here's an amino acid, generic structure of an amino acid, and this amino group is very toxic, so that has to get secreted in our urine. It gets converted into urea, and then it gets put into our urine, and it gets kicked out. So when we eat proteins, and we're metabolizing all the amino acids in a protein, the amine functional group gets tossed out. We got to get rid of it. Okay, here's an example of a freshwater protist called a paramecium. Okay, and it's at a big risk of taking on too much water. So it's got this organ called a contractile vacuole, which will actively pump water out of the cell. So that's the mechanism that this little thing has evolved. Another example of why it's important is you think about fish, okay, animals. Think about a fish that's in the ocean, okay, or a fish that's in a freshwater lake. Those are two very different environments, and the fish are going to have to respond differently. If you have a fish in the ocean, if we assume that's a high salt environment, we will consider that ocean to be hypertonic with respect to the fish. The fish is going to have certain characteristics. Okay, if you have a fish in a lake, we'll assume the lake is hypotonic with respect to the fish. Okay, so for a fish in the ocean, if the ocean is hypertonic, the cells of the fish are going to be losing water because they're trying to maintain equilibrium. They're going to lose lots of water, and the fish is constantly going to have to drink, and it is not going to pee very much. A fish in a freshwater lake, in a hypotonic situation, the cells of that fish will tend to gain a lot of water. It isn't going to drink very much water. It's getting it from the lake, and it's going to pee a lot. Okay, so it's super important to realize that homeostasis is at play, trying to keep our cells in a healthy, balanced situation. Okay, I'm going to talk a little bit more about homeostasis here, and then we will take a break, okay, for the next video. Okay, this is an example. Let's look at this graph for a second. Of blood glucose levels after a person eats. Okay, so the shaded area here is when they ate. So between 0 and 20 minutes, they ate some food. The black dashed line is the level of glucose that's considered isotonic or physiological, or when the body is in homeostasis. But the red line is what happens to the blood glucose levels after a meal. Okay, so we start out pretty close to where we need to be, normal levels. We eat, and blood glucose spikes, and then it starts to come down again. So something is going on that's causing this glucose to go up, and then causing these glucose levels to come back down, and then maintaining homeostasis. Okay? So it's very important for blood glucose levels to stay relatively constant over time. People that have trouble with that generally have diabetes or some other problems, okay? And there are a couple key hormones that our bodies use to maintain our blood glucose levels, okay? They are called insulin and glucagon. okay, and they're made in the pancreas. They're made in the hormone-secreting islet cells of the pancreas, okay, and those travel through the blood and they affect many different kinds of cells. Let's talk about glucagon first, okay. Generally, glucagon raises our blood glucose levels. Okay, so here's our stimulus. We haven't eaten for a while, we've skipped a meal perhaps, so our blood glucose levels go down, okay, that's the stimulus. The alpha cells of the pancreas are signaled to release glucagon into the blood, so that's our hormone. It goes to the liver, and it tells the liver to break down glycogen, which is the polymer of glucose, to release some glucose into the blood. Our blood glucose levels go up, and homeostasis is once again maintained. Okay, so glucagon is the hormone that gets turned on or released when we have not eaten. Okay, insulin, on the other hand, let's start here. Here's our stimulus. Let's say we just ate a big meal. Okay, our blood glucose levels go up because we just ate. Okay, that is the stimulus that signals the beta cells in the pancreas to release insulin. Insulin goes along and tells the liver to take up glucose and start storing it. Okay, because our cells have taken up more glucose. It starts to get stored, some of that extra glucose, as glycogen, and our blood glucose levels go down. Okay, so once again, our blood glucose levels get maintained. Okay, so this is just a recap. Again, in both situations, we have a stimulus and we have a response. We have a stimulus and we have a response. Both of those things are at play to maintain homeostasis, to maintain our blood glucose levels. It's important to note that these are both negative feedbacks. Remember, a negative feedback loop is when a response works against the stimulus. So here, the stimulus is low blood glucose levels. Oh, sorry, I said that backwards. High blood glucose, because we just ate. So that response is to lower our blood glucose levels. When we haven't eaten, the stimulus is low blood glucose levels. The response is to get, more glucose released, which is the opposite of the stimulus. Okay? So make sure you can answer those two questions. Which hormone is released after a meal, and which hormone is released in between meals? Okay, and the main guiding question then for this unit is, how do insulin and glucagon affect other cells and cause these things to happen? How does that work? Okay, that's the big question. We're trying to maintain homeostasis. Insulin and glucagon are just one example. Okay, and how does this happen? Okay, and this happens through cellular signaling, and I'm going to stop right here and make the next video.